Bismillahirrahmanirrahim. Assalamu alaikum everyone. My name is Dr. Sharjila and today I am here to talk about increasing the reproducibility of Chitosan nanofibers for biomedical applications. First of all, I would like to thank the ICPC committee who allowed me to present my talk here. First of all, let's talk about chitosan. What is chitosan? Chitosan is a sugar that is obtained from the hard outer skeleton of various seafoods. For example, crabs, lobsters. So these seafoods are popular around the world, and when their exoskeletons are thrown away after you can say getting the desired food from them, it can be used to produce chitosan. So we can say that chitosan is a material that is being obtained from a food waste. So that is why we can say that it is sustainable. Chitosan and its derivatives have found enormous applications in various industries. For example, in paper industry, in bioadhesive pharmaceutical, you can say chromatography membranes, cosmetics, food and feed additives as well. So our major focus here is the pharmaceutical or biomedicine industry. So as today we are discussing about the medical applications of chitosan, the medical textiles can be defined as fiber-based products or structures that can be used either as an first aid material or in clinical trials. Medical textiles are used whenever there is an accident or injury. So it can protect us from environmental pathogens and it provide a barrier against the microbes. So these microbes are present all over the skin and in the environment. So whenever there is a rupture in skin, these microbes find opportunity and they go there and colonize themselves and make the wounds infected. So that is the basic requirement of a medical textile, that it prevents from infection. So if let us suppose this is a healthy wound and when it becomes infected, it causes pus, redness, red streaks, and delayed wound healing, which results in non-compliance or patient discomfort. So we should avoid this discomfort. Therefore, whenever we got a wound, first of all, we try to sterilize the wound so that the surface is infection free, and then we apply some sort of medical dressing. Therefore, it will not be wrong to say that the first priority of medical practitioners is to avoid the infection or to clean the surface of the wound so that there are no microbes present to grow. So for this, you can say that if the medical practitioner achieves this, the wound heals properly without any scar or other complex issues and increases the patient compliance. So to avoid this infection, the medical practitioners often prescribe antibiotics. So in antibiotics, either these are given orally or through injections. So somewhere in our life at some point, every one of us would have been like eating these medicines for some sort of medical issues. It could be internal infection or due to some injury, and you know that these are associated with severe side effects including headache and upset stomach. But we can avoid this if we have some sort of wound. Instead of using this, if we use a polymer that has inherent antibacterial activity, for example chitosan, so these side effects could be avoided. So that is why chitosan is very important for biomedical textiles. So that increases the importance of chitosan for biomedical applications. I think now we are all clear why chitosan is important. So we can avoid various pharmaceutical agents if we are using a dressing of chitosan instead of eating the antibiotics. There are various methods to produce the dressings of chitosan that could be placed on various wounds. These include some traditional non-woven chitosan based dressings, or you can apply or coat a normal gauze with chitosan solution that could act as a good antimicrobial and hemostatic agent. However, my today's talk is about the nanofiber based chitosan dressings. So these nanofibers are very small in their diameter and they provide exceptional properties that are unmatchable when we compare it with conventional non-woven or other dressings. Why? Because they are excellent in air permeability. They allow gaseous exchange, as well as their smaller size hinders the passage of microbes to the wound bed. Therefore, these nanofibers are increasing in their importance and utilization in biomedical applications. Furthermore, one of the, you can say, important considerations to use nanofibers is that they are excellent mimicry of the natural extracellular matrix of the body. So if our skin layer has been damaged, so it has some collagen structure. You can see it is a 3D structure that promotes cell uh, proliferation and increases wound healing. So if we use nanofibers, these nanofibers are very near to the human extracellular matrix. That is why they can promote the cell attachment and could lead to enhanced wound healing. That is one of the, you can say, excellent feature of these nanofibers. However, it is not easy. There are various problems in producing nanofibers of chitosan. For example, they are diverse in their source of raw material. Chitosan from one source is completely different from another source, and it varies from region to region as well. So we can say that whenever we talk about biopolymers, there is a diverse range of molecular weight, purity, and other properties that could influence the reproducibility of the results. So for example, you can see that these are the nanofibers produced from two different origins of chitosan, and there is a big difference of diameter. These are very small fibers, you can see, and these are very big fibers. So the source of big fiber was the high molecular weight of chitosan, and these were produced from the low molecular weight chitosan. So these variations are not acceptable if you are trying to commercialize a product. So therefore, the reproducibility in natural polymers is a problem. So we have tried to reduce the problem by optimization of various parameters to become able to reduce the standard deviation as much as possible. That we will be discussing further. So for this we selected Toguchi based design for optimization. So we selected voltage, distance, flow rate at low and high levels according to the literature, uh, so that we can optimize the results and reduce the standard deviation as much as possible. Electrospinning is a simple, however complex process. There are various parameters that you can play with. For example, in solution you have concentration, viscosity, surface tension, conductivity, dielectric constant, and solvent volatility. So changing any of these parameters, you can have a significant influence on your fibers. Same, we have process parameters that are related to the machine, for example feed rate, distance, then the diameter of the needle. If you are using needle electrospinning, then electric field strength or voltage and electrostatic potential. So these processes are then optimized accordingly as well. And there are some ambient parameters like temperature, humidity, atmosphere pressure. So any sort of variation among these parameters could lead to a different results. So it is not that easy. You can say there are so many parameters that have to optimize to produce the same nanofiber with same diameter for reproducible results that can be commercialized. So we did a lot of study and lot of data is present with us that we optimize, and after optimization these like surface tension, conductivity, then we have selected the process parameters to find the smooth results. Before discussing the results, so first of all let us discuss some effects of these parameters on the morphology of fiber. So for example, solution concentration, voltage, flow rate, temperature, humidity, and distance. So as we increase the solution concentration, the diameter increases, and as we decrease the concentration, the diameter decreases. However, there are chances that if you are not at optimum concentration, you will have electrospraying instead of electrospinning. And the literature is contradictory regarding the applied voltage. In some papers you can see as the applied voltage was increased, fiber diameter increased. However, in others, the reverse is true. So this depends on the polymer and the interaction of polymer solution and other parameters as well. So this is, you can say, a bit contradictory. However, when we talk about the flow rate, so as the flow rate increases, fiber diameter, porosity, and bead defect formation increases. Why? Because you can see that the solution is not able to get polarized because the flow rate is higher, so there is less time for solution to get the charge. And when the flow rate is very low, you can see that there will be capillary tip obstruction due to premature solvent vaporization. So you can say that at lower flow rates, the solution will be uh, solidified at the surface of your tip, causing the needle choking. However, when we talk about temperature, there are other effects like the solvent vaporization rate increases, relative humidity increases the porosity, and the distance between tip can cause unstable electric field and process difficulties. And it is also one of the important parameters, the distance. The more the distance, the solvent can evaporate properly and you will have dry fibers. If the distance is, you can say, short, you can have a fused fiber surface, so that is not desired as well. So now you have an idea how these parameters are and what can be their potential effect on the morphology. So this is not easy, especially when you are dealing with chitosan and other alginates or biopolymers. That is why this study was important. These are the results of some of the Toguchi based experiments. You can see we have done various experiments, and these are the final diameters of produced nanofibers with their standard deviation. So here you can see the two SEM-MGs. So in the first image you can see this is a bigger fiber, and this is you can say smaller fiber. So this will increase the standard deviation. However, when we try to optimize at trial number 4, according to the design of experiment, the fiber diameter were very close. Only 29 nanometer deviation was observed. So this is quite acceptable. A deviation of more than 100 is common in nanofibers. So we have achieved less than 100 in almost every case. So we can say that we have found the exact parameters for that solution that cause the reproducible results or that reduce the standard deviation. So talking about the results, we can say that we have produced some exceptional reproducibility having almost 29 nanometer standard deviation, and this is very important for commercial aspects. Furthermore, these results were at lower percentage of chitosan. So if the percentage of chitosan is increased, the antibacterial activity increases. However, as the percentage of chitosan increases, its reproducibility decreases or standard deviation increases. So these are the opposite parameters where we have to compromise. For example, if we are requiring more antibacterial activity, we will add more chitosan, but it will cause increase in deviation and it will reduce the reproducibility and vice versa. So the smaller deviation is almost always preferable for commercial aspects. So let's talk about some conclusions. So we have achieved the lowest possible standard deviation so far. In literature we have seen standard deviation of 200 to 300 nanometer is possible. However, we have only achieved 29 nanometer standard deviation. So if we increase or decrease the molecular weight or concentration, the solution properties will change that and the results will vary. So it can be said that these findings are only valid for this study, this polymer, this solvent, this blend ratio and this machine. If you want to go for some commercial aspects and you choose some polymer of another molecular weight, then you have to first optimize. So as you are talking about production and higher productivity, you can once optimize your parameters and results, then you can go for commercialization. That will become very easy. So with this, my time is complete. That is 15 minutes. So if there are any questions, you are more than welcome to ask, and you can always email me at shajil.habib@ntu.edu.pk. And we have many facilities and working in collaboration with various universities and stakeholders. So if you have some interest regarding research or commercial activity, you can always reach NTU. So not only me, you can reach anybody in NTU and you can work in collaboration.